Все.
Писать как?
Приходите.
Всё,
Можете
спасибо.
прерывать.
Good afternoon, students. Today we speak with you about non-Mendelian inheritance. All of us studies genetics during of medical biology course, and you know which one Mendelian inheritance refers an inheritance pattern which follows two Mendel laws. Mendelian inheritance refers to patterns of inheritance which are characterized for organisms which produce sexually. And the Mendelian inheritance provide the inheritance of genes according of third Mendel laws. Huntington disease, hemophilia, cystic fibrosis, all these disorders are inherited according Mendelian inheritance, and we can do the inheritance of these diseases in different generation by using of Mendel rules of consequence of single gene. So the Mendelian inheritance is explain the inheritance of single gene disorders with complete dominance. But the scientists found which were many human traits. The scientists think near of 70% have more complicated modes of inheritance than Mendel traits. For example, the non-Mendelian inheritance is characterized for traits with codominance, incomplete dominance, polygenic traits, and others. So a non-Mendelian inheritance refers the inheritance of traits which have a more complex genetic basis than one gene with two alleles and complete dominance. And today we speak with you about genetic polymorphism. The polymorphism is a term used in genetics to describe multiple forms of single gene which provides which are present in individual or among a group of individuals. Instead, polymorphism refers to forms which are discontinuous. It can be bimodal or polymodal. For example, ear lobes are if attached attached or not. So it is an error or trait. So polymorphism. These are mutation in genes which provides the common nucleotide sequences in population. The polymorphism we call the mutation the polymorphism the mutation, the frequency of which in population near of 1%. The monomorphism, this is part of DNA where the nucleotide sequences is the same for every one population. The polymorphism plays very important role in evolution because each provide the variation of characteristics in organisms of population. So polymorphism and many traits display a variation within population. The polymorphism provides the material for evolution, and each provide the high level of genetic diversity in populations. The genetic polymorphism refers the occurrence of two or more genetically determined genotypes in certain population in proportion which the rest of the characteristics not be made just by recurrent mutation, general frequency of mutation. Polymorphism promotes diversity and persist over many generations because no single form has an overall advantage or disadvantage over the others in terms of natural selection. So the reason of genetic polymorphism are population. A polymorphism is a DNA sequences variation which is common in the population. In polymorphism, there are two or more equally acceptable alternatives. To be classifies a polymorphism, the least common allele must have a frequency of at least 1% in the population. If the frequency is lower than this, the allele is regarded as mutation. So students, we call the mutation by genetic polymorphism if the frequency of this mutation is more as 1% in population. If the frequency is least as 1%, we call these changes of DNA the rare mutations. For example, if the leaves on a plant were normally green, with varying shades of red veins, and a leaf was found with yellow veins, each could be considered a mutant if less than 1% of leaves of which phenotype had yellow veins. Otherwise, it would be considered to be a polymorphic trait. So, as a result of this, you should understand which changes of DNA do we call polymorphic traits. Only if the frequency of these genes more as 1% in population. Polymorphism involves one of two or more variants of particular DNA sequence. The most common type of polymorphism involves variation at single base pair. Polymorphism can also be much larger in size and involve big stretches of DNA. И как-то вышло совершенно. Ко мне вернулся аппарат.
Это
Да. А можно ли, чтобы вот чуть-чуть оно было поменьше? Просто мне не все видно. Вот так. Спасибо. So students, the changes in single base pair has name a single nucleotide polymorphism or CNP, pronounces SNP. Scientists are studying how CNPs in human genome correlate with a disease, drug response, and other phenotypes. Polymorphism, by strict definition, which hardly anybody pays attention to anymore, is a place in the DNA sequence where there is variation and the less common variant is present in at least 1% of people of who you test. That is to distinguish, therefore, polymorphism from a rare variant that might occur only 1 in 1,000 people. A polymorphism each has to occur at least 1 in 100 people. Polymorphism could be not just single letter change like C instead of P. They could also be something more elaborate, like a whole stretch of DNA that is either present or absent. You might call that a copy number variant. Those are all polymorphism. But this is basically a general term to talk about diversity in genome in species. Gene sequencing studies like that done for Human Genome Project have revealed that at the nucleotide level, the gene encoding a specific protein can have a number of differences in sequence. These differences don't alter the overall product significantly enough to produce a different protein, but may have an effect of substrate specificity and specific activity for enzymes. Also, an effect could be binding efficiencies for transcription factors, membrane proteins, or other features and functions. For example, within the human race, there are many different polymorphisms of CYP1A1 of many cytochrome P450 enzymes of the liver. Although the enzymes are basically the same sequences and structure. Polymorphism in this enzyme can influence how humans metabolize drugs. CYP1A1 polymorphism in humans have been linked to smoking-related lung cancer due to the prevalence of certain chemicals in cigarette smoke. Polycyclic aromatic hydrocarbons, which are metabolized into cancerogenic intermediates, the product of the process. There are different changes in alleles and they provide the different phenotypes. Here you see the examples. So on screen you can see the common pharmacogenetic polymorphism. So the polymorphism in cytochrome P450 causes the different answer of people to using of different medicines. So you can see which polymorphism provides, how does the polymorphism provides the speed of destroying of substances in our body. Uh, there are many types of polymorphism. This is a restriction fragment range polymorphism, tandem repeats, single nucleotide polymorphism. So single nucleotide polymorphism, this is single base differences in population. Microsatellites, short tandem repeat. This is 2-6 base pairs long. Mini satellites, simple sequences repeat 10 from 10 till 40 base pairs long. Variable number of tandem repeats, the number of repeats there are many number of repeats and they may vary. Restriction fragment large polymorphism, presence or absence of site. Deletion, duplication, insertion, these are alteration on chromosome level. And complex haplotypes, combination of the above. So now I tell you about single nucleotide polymorphism, SNP. These are positioned in genome where some individuals have one nucleotide and others have a different nucleotide. For each SNP could potentially have 4 alleles because there are 4 types of nucleotides. So we have 4 variants of this type of polymorphism. This is single nucleotide polymorphism, and here you see the short tandem repeat polymorphism. The short tandem polymorphism we can divide in 2 types: microsatellites, satellite, and mini satellite. Microsatellite, these are short tandem repeats which consist of 1, 2, 9 base pairs monomers repeating sequences. Mini satellites, these are tandem repeats which are monomer repeat length of 10 to 100 base pairs. Microsatellites rich with A adenine and thymine bases, and mini satellites rich with guanine and cytosine bases. The microsatellites, also known as simple sequences repeat (SSR) or short tandem repeats (STR). The mini satellites, also known as variable number tandem repeats VNTR. So the key difference between mini satellite and microsatellite is the size or range of repeated sequences. Restriction fragment length polymorphism RFLP is a type of polymorphism which results from variation in the DNA sequences recognized by restriction enzymes. A restriction enzyme cuts the DNA molecule at every occurrence of a particular sequence called restriction site. For example, Hind enzyme cuts these sequences. Any mutation of single nucleotide may destroy or create the site. For example, AACCTT instead of AAGCTT for Hind free. RFLP analysis is the detection of the change in the length of restriction fragments. A haplotype, haploid genotype, is a group of alleles in an organism which are inherited together from a single parent. Many organisms contain a genetic material which is inherited from two parents. Normally, these organisms have their DNA organized in two sets of pairwise similar chromosomes. The offspring gets one chromosome in each pair from each parent. A set of pairs of chromosomes is called diploid, and set of only one, half of each pair, is called haploid. The haploid genotype, haplotype, is a genotype which considered the singular chromosomes rather than the pairs of chromosomes. Each can be all the chromosomes from one of the parent or a minor patch of a chromosome, for example, a sequence of 9,000 base pairs. A haplotype is in its most general sense referring to a set of DNA variation along a chromosome that tend to be inherited together because they are very close together. They get inherited together because they not generate crossovers or recombination between these markers or between these different polymorphisms because they are very, very close. So a haplotype can refer to a combination of alleles in single gene, or it could be alleles across multiple genes. It could be single nucleotide polymorphism that are not in a gene, but are in between genes. Basically, it just means that these are variations in DNA which are so close together that they tend not to recombine and therefore tend to be passed down through the generations together. And the International HapMap project has given us a very excellent tool to detect these regions of haplotypes which are passed together and to use those in genetic studies. So students, it is very easy to tell about haplotype of Y chromosome because the most part of Y chromosome doesn't have homologous parts in X chromosome. So the most genes of Y chromosome move from generation to generation together. Now since we tell about trinucleotide repeat disorders, also known as microsatellite expansion diseases. These are set of over 50 genetic disorders caused by trinucleotide repeat expansion, a kind of mutation in which repeats of three nucleotides repeats increase in copy numbers until they cross a threshold above which they become unstable. Depending on its location, the unstable trinucleotide repeat may cause defects in a protein encoded by gene, change the regulation of a gene expression, produce a toxic RNA, or lead to chromosome instability. Generally, the larger the expansion, the faster the onset of disease and more severe the disease become. Here you see the examples of trinucleotide repeat expansion. So you see in first column the repeated sequence. You see in second column normal range of these repeated sequences, and in a third column you can see the disease range. So you see which the number of copies increases in a disease condition. The most common diseases these are Friedreich ataxia, spinocerebellar ataxia, myotonic dystrophy, and Fragile X syndrome. One of examples of trinucleotide repeat expansion is Huntington disease. This is neurodegenerative genetic disorder. The reason of this disease, this is a trinucleotide repeat of cytosine, adenine, and guanine. Each happens in short arm of chromosome number 4. This is autosomal dominant disease. So, in affected people, the number of repeats increases from 36 till 121, and the normal number of repeats from 10 till 26. The permutation stage is from 27 to 41 repeat. So the reason this is increasing of CAG repeats from 36 till 121. As a result of this mutation, the polyglutamine number increases in human Huntington protein. Each causes the misfolded proteins and they cause death of neurons in brain and muscle cells in the body. Scientists have known that in people with Huntington's chaperone proteins, whose job it is to fold misfold proteins are at low levels, but it wasn't clear why. So, as a result of this mutation, the death of neurons happens in the brain. The disease mostly starts after 30, 40, 50 years old, and the main symptoms with chorea, depression, anxiety, trouble swallowing, problems with concentration, balance problems, slow eyes movement, and many others. The people with this disease lose the psychomotor control of the body. The next disease is Friedreich ataxia. This is genetic disorder which caused by a defect in the gene frataxin, which causes their body to produce too much of part of the DNA called a trinucleotide repeat, GAA. The affection of brain and spinal cord happens, and also the heart affect too. So the reason of this disease, this is increasing of number of GAA repeats in intron of gene of frataxin. So the number of proteins, the number of this protein decreases. These are the symptoms of this disorder. This is affection of spinal cord and brain, This is weakness of muscles. This is a distal sensory loss, so many symptoms of this disease. Хорошо, да, я сейчас перейду на то.
Сюда.
Хорошо, я поняла. Okay, students, we continue the lecture. So you see the symptoms of Friedreich ataxia. The specific symptoms of this disorder with damage of spinal cord and brain, and each causes. Все, спасибо. And the weakness of muscles happens as a result of this. The kyphoscoliosis is observed in these patients and high arches, pes cavus. The next genetic disorder is Fragile X syndrome. Fragile X is a genetic disorder which is caused by break or weakness of the long arm of X chromosome. The frequency of this syndrome is 1 case to 5,000 newborn children, and the males affected more often as females. Is the second most common inherited cause of mental retardation due to chromosomal abnormalities. The specific symptoms of this disorder is mental retardation, large ears, prominent jaw, and pale blue irises. The reason of this disease, this is increasing of number of trinucleotides CGG. For disease, the number of these repeats should be more as 200. The syndrome is called Fragile X because there exists a fragile site or gap at the end of the long arm of the X chromosome in lymphocyte of affected patients when grown in a folate deficient medium. The common physical features of this syndrome, these are prominent broad forehead, large ears, long face, mental retardation, murmur mitral valve prolapse, hollow chest, hypotonia, scoliosis, and in male macroarthritism. Plus mental retardation, which can be autism spectrum disorders, intellectual disability, distinct facial features. Now, students, we tell with you about genomic imprinting. It is thought that the maternal or paternal imprint is erased with each succeeding generation. A male receives a maternally imprinted and paternally imprinted chromosome, but will always transmit a paternally imprinted chromosome 15. Note the maternal-paternal imprinting is heritable through mitosis. So, genomic imprinting includes epigenetic differences, result in differences in expression and DNA methylation. This is a reversible chemical modification of DNA. The literally epigenetic means above or on top of genetics. Usually this means information coded beyond the DNA sequence, such as in covalent modification to the DNA or modification to the chromatin structure. Sources of genetic changes which can produce disease: mutation in DNA or uniparental disomy. Epimutation, this is abnormal chromatin formation at 1 plus loci in the genome. Primary epimutation, immediate cause of disease through the DNA methylation or histone acetylation. Secondary epimutation controls epigenetic processes. The epigenetics is very important for differentiation of cells of our body because all cells of our body contain the same genes, but gene expression patterns are different in different cells. So as a result of this, the differentiation of cell happens. The role of epigenetics has been known for many years. We study with you the Lion hypothesis. This is inactive X chromosome in female cells. So inactive X chromosome is heavily epigenetically modified. And we can find this condition in female bodies. So the epigenetic mechanism can regulate genes involved in differentiation, cell cycle, and cell survival. Deregulation of epigenetic mechanisms result in aberrant gene expression, which can lead to cancer. Reversal of deregulated epigenetic changes is a rational strategy for targeting cancer. So during of epigenetic, the changes of expression of the epigenetic, these are changes with a mechanism which provide the different expression of genes. The genes can be silenced, prevention of gene expression, enhanced, increase of gene expression, and changed, transmitted to next generation. Some changes can be reversed. The environmental factors can change the expression of genes. The most epigenetic modification, epigenetic marks that are established in most tissues during an organism lifetime are irrelevant with respect to next generation. This is because epigenetic modifications are normally erased at each generation. For example, DNA methylation is typically removed or possibly oxidize during zygote formation and re-establish through successive cell division during development. And also because only epigenetic modification of the major gametes and not in other tissues have the potential to contribute to the phenotype of the spring the next generation. Only those epigenetic modifications which are heritable via the gametes germlines lead to epigenetic inheritance. So the epigenetic can be control for gene expression can be provided by histone modification. Acetylation, methylation, and phosphorylation affect the genome in different ways from one region to another. How much it loops and coils, activates or deactivates of transcription. Generally speaking, acetylation loosens the chromatin structure to make genes available for transcription. Deacetylation causes the chromatin to coil tightly and silences transcription. So the gene can be switched on or switched off by acetylation or deacetylation of histones. The other way of regulation of gene expression, this is methylation. This is addition of methyl group, carbon H3, to the cytosine bases of DNA. Silences transcription and replication of transposable elements. Methyl groups prevent the binding of transcription factors with cytosine. Transcription cannot be initiated, so the methylation prevents the transcription of this gene. So during of methylation, the 5' position in cytosine can be methylated by DNA amethyltransferases. Some DNA methyltransferases act during DNA replication to maintain methylation patterns heritable. Aves de Novo. So the ways of epigenetic alteration, this is methylation of DNA, modification of histones, and RNA-mediated modification. Uniparental disomy, presence of two homologous chromosomes, inherit from only one parent. Isobisomy, parent passes on two copies of the same chromosome through non-disjunction in meiosis II. Heterodisomy, parent passes on one copy of each homologous chromosome through non-disjunction in meiosis I. This non-disjunction can cause the trisomy, uniparental disomy, or normal organism. Examples of diseases affected by genomic imprinting. This is Prader-Willi syndrome, caused by regional deletion or mutation in paternally inherited chromosome 15. Engelmann syndrome, a different disease phenotype caused by regional deletion or mutation in maternally inherited chromosome 15. Both of these disorders are caused by deletion of a region of chromosome 15. But Prader-Willi syndrome is caused by deletion in paternal chromosome, and Engelmann syndrome is caused by deletion in maternal chromosome. This syndrome have the different features, different symptoms, different affected organs. Prader-Willi syndrome, obesity, mental retardation, short stature, Engelmann syndrome, uncontrollable lava, jerky movements, and other motor and mental symptoms. Syndrome that develops depends upon the parent which provide the mutant chromosome. Engelmann Prader-Willi syndromes commonly referred to as genomic imprinting. Imprinting is not the cause of this syndrome, but it's responsible for unacute presentation of this disease. So, the genetic mechanism of Engelmann syndrome, the deletion in long arm of maternal chromosome 15 happens. Prader-Willi syndrome. The infants with this syndrome are very weak because the sucking reflex is poor. So the growth is poor too. Other phenotypes associated with the syndrome includes poor sexual development in males, behavioral problem, mental retardation. Many individuals with this syndrome are undiagnosed, so the frequency of this syndrome is not known. The four known genetic mechanisms can cause the Angelman syndrome. The 70% of Angelman syndrome cases result of deletion of maternal chromosome de novo. So this deletion occur in ovum during the gametogenesis. Approximately 2% result from paternal uniparental disomy, and 2 to 3% result from imprinting defects. A subset of remaining 25% are caused by mutation in the gene encoding the ubiquitin protein ligase. This is Angelman syndrome on pictures of Italian painters. We have mental retardation, small chin, uncontrolled laughing, neurological problems, and others. Okay, students, the lecture is finished. Goodbye.